Here we go. Hey, today on the Soil Talk podcast from Elevate Ag, we have Zach Wright from Living Soil Labs in Canton, South Dakota. Is that correct?
Yeah, a living soil compost lab, but
Okay.
that
Yeah.
works too.
Well, I've known you, Zach, for a while. You helped me through the soil food web school a little bit, and some of my questions that I had when I took that class. And you've known that community a long time, been a key player in that community for a long time. So we're just really fortunate to have you today. Uh,
Thank you.
our topic that I wanted you to discuss is the importance of fungi in our soil systems, a healthy soil system.
hmm
And then we'll go on from there as to what we can do to enhance that and maybe what we should be doing to keep it healthy and keep things functioning. So I'll let you take it from there, Zach. I mean, what first of all, what is fungi and where does it fit in a healthy soil?
Well, the way I like to start things off is really reminding us that the soil is the gut. It's the gut of the system. And just like our own guts, we need a balance of flora and fauna for that gut to function. And ironically, I just bought this book yesterday and I've already read the entire book. I stayed up all night reading it, and it's called, oh boy, The
Yeah,
Gut
you're
Brain Paradox by Dr. Stephen R. Gundry. And it's really fascinating because so much of our illnesses and diseases, as we go deeper into the science, we're finding that it's due to our gut issues. It is really, as he describes, as he says so poignantly in this book, death really does begin in the gut. And but so does life. And we can actually turn around a lot of our gut issues with probiotic means. And what I mean by probiotics is intentional biology, the act of actually putting the biological components back into the system so the system can work again. So back to fungi. Fungi is a critical component to the soil, aka the gut of the system. Fungi, in other words, is the other half of the bottom of our food chain. The other half being bacteria, which every soil system that I've ever analyzed has, the other half being fungi. And what, Dr. Elaine Ingham's work and other soil ecologists' work have indicated is that the crops, the cash crops that we want to be growing, we really want to be seeing as close to an equal biomass of bacteria and fungi in those systems. So, in other words, we don't want an excess of bacteria in the system and a lack of fungi, or the system is dysfunctional. We don't have a system that's actually functioning correctly. And so looking at the soil like a gut, we are also, as agrarians, we're starting to see the importance of repopulating the soil with the type of organisms that make this system function again and digest again. And why fungi is important, one of the main things is it decomposes things that many of the components of plants that bacteria can't. Fungi came along a lot later than bacteria did, and fungi came along when your angiosperm type plants started coming around, and this was, a long time ago. They say the Paleozoic era, so about 200 million years ago, give or take. I'm not here to say that's right or wrong, but when we look at fossil records, we start to notice that as plants started to have a more lignified, stronger cell structure in the plant itself, fungi started appearing in the fossils. So there was a coevolution that happened where fungi was symbiotically, naturally required to grow with these harder skinned, woodier plants over time. And why was that function there? To digest and decompose those types of plants to return them back to the soil to beget more fertility over time. And that's why we see higher fungal components in our woods, in our woodlands, our forests, our undisturbed areas where we have woodier plant species, is because we have a live, teeming fungal community underneath the soil versus most of our conventional farm crop acres where we have a very bacterially dominant system. lacking fungi. And, working with different decomposing, crop residue type trials and programs over the years, I noticed things like smart stack genetics are really hard to decompose. It's like they have been designed to withstand abiotic stress, aka weather, so that the stalk actually stands up and we don't have green snap and we have a stronger plant. But it's like a consequence of creating that stronger plant with a stronger cell wall and perhaps higher lignin in that plant material, the the plant cannot decompose very easily. And in fact, in looking at decomposing stuff on Smart Stack residue, we noticed that it just seems to take a lot longer for a biologically diverse, high fungal culture that we spray into that material to actually break down. So I think this is an issue. I think that ultimately that trash is, as a composter, that trash is actually food. And waste is food, you could also say. Something is ultimately going to eat it.
And in a
So,
corn crop, it's next year's nutrients in some sense, right? You're
exactly.
bringing it back into the system.
Exactly. And so fungi, very important. The other half of the bottom of the food chain. And just starting there when it comes to the succession of trophic levels of organisms. So the things that eat the things that eat the things before we could start to see it with our naked eye. Fungi is so critical because it's at the bottom of that web, that soil food web, you might say. So as the farmers that I work with that are seeking a least cost producer status or a organic or a regenerative sort of model, a sustainable model, we are taking dollars out of the fertilizer system and the chemical system, and we're investing more of anything our time into creating these fungal as well as other organisms, these living components that we're actually putting into the system in hopes of actually digesting the residue and turning it into soil again on an annual basis. I would say over the last 13 years of doing this, the awareness, um, the clue of, hey, what can we actually spray in the fall on our residue to actually make it into soil again is growing. I had a lot more people this year asking me about and you know just kind of kicking the tires. What can we do post harvest? A lot of people got done early this year. So what can we do post harvest to benefit next year? And it's like, well, compost right in the field, turn that trash into food again. And by putting the organisms into that trash food material, we're composting right on the site. And yes, it's going to take time. In years past, I like to throw these things out there so people don't have this I don't know. I'm not trying to paint everything with rose-colored glasses, but you're not going to see it in March in what you applied necessarily. I've had one anecdote where somebody noticed their soil or their snow melted quicker where they applied their biology versus the check strips where they didn't apply biology. But I would say by June, you're going to notice your crop residue where you've applied it is much more reduced, if not fully disappeared. And that's because you've put the organisms back into place to break it down. And one of those key organisms to breaking down that residue material, that highly lignified material, those cell walls of the plant that are left laying on the field, is fungi. How do we get fungi? We make it, we culture it. There are products out there. There's foundational fungi, which is a wonderful culture that is very high in fungi that exists on the marketplace that you can purchase. Um, I work with a number of farmers that want to make their own fungi, which you can culture yourself. You can use fungal products like foundational fungi or, say, soil from your forest itself, and you can actually put it into your own culture that you make and essentially grow your own fungus that way. And then we literally wash it. We extract it. We make a liquid out of it. And with filters in place, we apply that out into the system at any rate. We can put it through a sprayer. We put it through center pivots down in Nebraska and Kansas. We inoculate the system with what is ultimately missing in the system. Now, why do I say, why is it missing in the system? That's because fungi is very sensitive. And for a long time, I would argue that every organism in the system was sensitive. And I've seen a lot of things through the microscope that have kind of turned me upside down on that, where, commercially smart stack treated seed with biology on it actually didn't kill the biology that we put on it, and we verified that through the microscope. But fungi is one of those things that I notice really takes a hit whenever we apply our post herbicides. I think it takes a hit from our pre's as well. I think fungi also and my perspective is purely through the microscope. So I've using binoculars essentially, or a telescope, to look really close at what the life forms actually are are either there or not or doing. Um, Commercial fertilizers also heavily disturb fungal component in soils. Now, to compare another disturbing event like tillage to a chemical spray, this would be my analogy. And this is again based on direct observation. Tillage is like an earthquake. It's going to absolutely turn everything upside down. But strangely enough, and I'm talking in this context, a mold bore plow experiment we did. We turned that whole food web upside down. So we went from a 5-3 fungal, 5 times fungi to 3 times bacteria, to like a 1-30 fungal to bacterial ratio just by mold bore plowing. But what we found was that the fungi was still there. So were the protozoa, which are second rung of the food chain. So were even our nematodes were still there, but our bacterial growth went through the roof. So we created this event that created this earthquake in the system. A chemical pass oftentimes is more like a nuclear bomb. So what I observe post chemical passes, and when I say chemicals, herbicides largely, because in the wonderful no-till systems that, and I don't say that in jest. I mean, no-till, it has its place for sure. But when I hear no-till, I hear chemical. And when I see a lot of these soils right after they've applied their chemical, having looked at it before, I noticed that that fungal component, especially, it's like it's just fried. And what is left over, it's like the bacteria have adhered to the hypha, the actual threads that we see in the microscope itself, and they're consuming it, they're eating it. Our protozoa gone, our nematodes gone. So it's that in that sense, you were managing disturbance. So if you're an organic person who's tilling or you till anyway, you're going to be slicing and dicing your fungi. If you're a no-till person and you're applying chemicals, you're going to be frying a lot of your fungi. So the takeaway is we want to ultimately, and I'm not telling anybody what to do. I've been definitely accused of that, but as an inspiration, as an idea, we want to potentially consider planting microbes, this highly diverse culture of organisms that have specific functions that can help the plant, help our soils digest in so many different ways, create more organic matter, create more air capacity and water holding capacity. These organisms are carbon forms themselves, but the idea is why don't we consider planting those with our seed? And thus we treat our seed, we do a seed treatment, like a commercial seed treatment, with these cultures of biology containing specifically fungi. We also put this stuff in our in-furrow or liquid in-furrow. We apply these organisms around the seed in hopes of not only protecting the seed against pathogens and things like that could be lingering in these early cold spring, wet spring soils, but we're hoping that we grow the biology with the plant. And that's what we see oftentimes, especially when we go out into the field, even just I mean, a week, but I've seen 48 hours after that seed's been put in the ground. That very first chit that comes off of that root that shoots off of the seed is absolutely covered in soil. It's like it's stuck with soil, and that's called the rhizosheath. So that's one way that we can analog what's happening out in the system, that feedback loop that we can check out in the system to see if our plant and our soil are talking. We can go dig the roots up of the plant at any stage. And I'm telling you, like even after 48 hours and it's just sprouted, we can pull that sprout out of the ground and we can see whether or not our biology is there in a really gross general way of saying by if we see that soil material stuck to the root itself and covering it kind of like a dreadlock. And that's just kind of one soil health principle that we can use out in the landscape without any real tools other than a spade to maybe dig the plant up to monitor the biology, the soil microbiology plant interaction without having to look at it through a microscope.
Is it possible, Zach, and from what your experience, okay, the guy's gonna have to go out and spray his weeds. Got a big mess in a no-till situation. I gotta spray my weeds. And then can you come back and reestablish that population effectively after, maybe, give it a week or 10 days and get a foliar pass to reestablish some of that?
So to the extent of reestablishing, that varies depending on the chemical. I have done some preliminary work as commissioned by farmers that had these 10 chemicals that they 10 different chemicals, right? With skull and
Hmm.
crossbones on them. Like be careful with these things. But these 10 different chemicals that they were going to use this year, and they said, let's conduct a trial on this soil and see what our soil microbiology populations do with these 10 chemicals. And so I've done double blind studies looking at that, and not all chemicals are the same. But to answer your question, the easiest way to I guess look at it and what I see with the majority of my clients that I help kind of initiate this whole program of biological measure and replenishment into the system is we want to manage disturbance. So if we've sprayed said chemical, regardless of knowing how bad that chemical is or not, let's follow up with the cleanup crew, the bioremediation crew, and that's your biology. And so typically I say allow the mode of action of that chemical, give it a solid 3 to 7 days to work, to absorb into the plant, to target and eliminate the thing that it needs to eliminate, and then let's replenish the system again. Now does it get back to where it was prior to that application? That really varies. Pre-emergent herbicides, I mean, I've seen some glorious looking soils in March and April that after the pre's go in, just absolutely tank. And it's like
Hmm.
it isn't until July and multiple applications that we start to verify that, okay, our biology is functioning again. And how am I basing biology as functioning? I'm looking for the trophic levels. I'm looking for the bottom 3 to 4 rungs of the food chain being in place because then I know we have things that are eating things that are eating things and ultimately cycling and creating these postbiotic, these secondary metabolites, these enzymes, these humic substances that are very plant available and plant critical for its use. And in a sense, this is all about creating more humus. And humus is a very, oh, what's the word I'm looking for? It's a very ambiguous word. It's something that we've been looking at for hundreds of years in science and trying to really understand what is this substance? What is this dark brown aggregated cottage cheese substance that we can go into our forest, remove some leaves, and literally grab with our hands out of the dirt? What
It's
is
like
this material?
define human.
Right. Exactly. Exactly. It is stabilized carbon. It to
Yeah.
me, it is the mark of digested carbon material, material that has been broken down and eaten. And as I was reminded in this gut brain paradox, in our guts alone, there are bacteria that eat the prebiotic, the food, the thing we've digested, and there's bacteria that come along and eat those bacteria, and bacteria that come along and eat those bacteria, and constantly are creating these postbiotic materials that eventually get to a point of stability that our body can use them. And
Mm.
I think that's essentially what we could say is happening in our soils too. And I know that's a very general thing to say, but it's not lost on us when we are doing DNA analysis of soils, and we're seeing over 4,000 kinds of bacterial and fungal species alone in that soil. So there's a lot of players at play in these systems. And one other thing that I want to say real quick is we know that we're kind of enslaved in a sense to the monoculture. We're planting this one thing, that's our cash crop, and it's very susceptible to nature, especially when it's weak or it's sick. This biological measure that we're taking, that we're spraying and we're injecting and we're applying into our systems with these organisms that I'm talking about, that's your polyculture. So we can promote a polycultural system underground that in a sense helps protect our plants above ground. Now that's not a foolproof, like you're not going to have any insect pressure or anything like that. Western bean cutworm is a terrible insect. Um, but we can mitigate a lot of the stress of the plant by having the organisms themselves in the system digesting last year's residue, perhaps even the year before that's residue, and creating those humic substances again that act as batteries in our system, allowing for nutrients from our sand, silts, and clays, our parent material, to become available again. It's said that fulvic acid alone, that humic substance that is very easily made through any sort of aerobic composting measure, fulvic acid alone makes iron and silica, it strips it, it peels it like a layer from an onion off of your sand, silts, and clays just by its very nature. So, literally, that humic substance of fulvic acid is weathering dirt, it's weathering the actual minerals in our soil and making them plant available again, acting as kind of a battery.
So this whole discussion, I mean, it comes into today. You can't hardly turn on the news without hearing about, what are the farmers going to do? The price of fertilizer is going up and the prices of commodities is down. And, there's kind of this upside down economy out there right now. Well, if we're going to even try to stay in this game, I mean, how much of this we can get working for us is to our advantage, is it not? I mean, there's still going to be some input costs, but you're working with your own system, your own regenerative system where you aren't having to bring all these off-farm inputs.
Right. Right. This tends to bring out this sort of mindset and program for the guys that I observe, the dozens of guys that I observe, and that's a conservative figure. I mean, there's a lot of people doing this more and more every year, and it's wonderful to see. Um, it brings out the least cost producer. So even though I busheled 250, 250 bushels of corn, I had, 240, 250 3 years ago. I spent so much less money because my one of my main inputs was this biology, this biological thing that I'm either making myself or I'm sourcing it. There are a few companies out there that are making this liquid biological program, which is how I've met a lot of Elevate Ag customers is because people have contacted me saying, "Hey, I tried um, Elevate Ag's liquid biological kind of extract product with that foundational fungi in it. And I saw great results. Can you explain more about what happened? It's just, I field those calls all the time. People that are like not ready to drink the Kool-Aid, but they've tried something and they've said, wow, I noticed a big difference not necessarily in the yield, but in the plant health, and I used 20% less nitrogen, or
Right.
I completely cut out P and K, but this input allowed me to do so. How did that work? And that's because these life forms themselves are creating phosphorus. Fungi especially is like a phosphorus pipe. And even in some of our texts that go back over 100 years, they were cautioning us against using orthophosphates because it would retard I mean, that's how it was described. It would subdue the natural fungi in our systems. And they were talking about mycorrhiza fungi over 100 years ago in text, how the use of synthetic phosphorus fertilizers would not only make our fungi that are inherent in our soils not work, but it would also harm the fungi itself. So, as a teaser, I have seen countless number of people now quit pee by going down this biological path with that fungal component verified in the material itself. Much of what I do as a lab is I help farmers verify that they are getting that fungal component in their product. So I look at a lot of products. I'll go to a farm and all of a sudden I've got 10 2-bys on my table and they just want me to shake it up and blind draw and tell them what I see in the microscope. And most of your bug in the jug products out there, I'm sorry to admit, don't have any sort of noticeable fungi in the product. So having a verified fungal product is absolutely paramount to this biological thing that we're doing. Not that these bacterial products aren't beneficial. There's definitely a utility to some of these cultures of 10, 15, 20 kinds of bacteria that fix nitrogen or perhaps even fix phosphorus. But I think if we're looking at the long game, like you were saying, Jim, we want to be looking at the bigger picture and we want everything to grow in totality. So, in my easiest summation, and this is how I would describe it to a fifth grade class that I would be talking to, we're trying to put everything out into the system so everything can grow, not just the plants, but we want the soil to grow. And in order for the soil to grow, we want to replenish it with the bacteria, the fungi, the protista, the bottom couple rungs of the food chain that ultimately are getting hammered by our modern agricultural practices.
And we go down this path, and if you start getting this system working and you're saving a little money, there's a whole bunch of other downstream environmental benefits that begin to flow in as well. Like, I mean, you're not losing nutrients to your waterway anymore. You're not going backwards on your organic matter. It's the biology that can build it.
Yeah.
It has to be that way. It
Oh, yeah.
has to go through biology to get higher organic matter.
It's a
And
refinery, the
so we're bringing
biorefinery.
life in and just giving them the tools and then get out of the way. It's the way nature would do it.
And one of my favorite things to observe is in this transition, because again, I'm kind of the boots on the ground that can really, I think, instigate, because I've done this long enough now to really kind of instigate this whole biological component and how to get it into the system at a very rudimentary level. And I would say one of my favorite things to witness is all of the anecdotes that come out, even year one. And those anecdotes aren't necessarily, I had the greatest yield I've ever had. I mean, in fact, I'd put $20 on the table that in the first year, you're not going to have the greatest yield you've ever had because ultimately, this is a long game transition. We have to replenish these organisms into the system that we're continuing to pound with fertilizer, with chemicals, with tillage, with high levels of poultry litter. That goes out to you organic guys out there that are very salty, very, very bacterially dominant, almost bacterially exclusive. And so we're trying to balance that bacterial biomass by bringing more fungi into the fold. So I like to put it this way. It's kind of like an initial 3-year commitment to, okay, I'm going to apply this biology every year. I would ideally like to do it multiple times a year. So we try to target what is the critical times, depending on the equipment that's available and the system that we can put that biology in. And we give it 3 years. But even in year one, the anecdotes start coming. I have more worms. I see worms. I've never even seen worms before. Earthworms, night crawlers in my system. Where the heck did they come from? I have more birds that are eating my insects in my field. That's an amazing sort of thing to observe. I have mushrooms growing in my understory. I've never seen mushrooms in my field before. Um, one of my customers, a longer term customer now, one of the things we saw in year two, which was interesting, and for context, this would have been around Marshalltown, Iowa. His field abutted to his neighbor's field, and both of them had drain tiles just about 12 feet apart from one another coming from their fields. Very different practices. Now, this farmer that I was working with was doing things very biological and using a lot of foods as his fertility, a lot of amino acids and sea minerals, things like that. The neighbor, absolutely standard, high amounts of 32, high amounts of fertilizer in general, MAP. Um, he was really pounding it. One thing we saw was his the farmer that was doing biology's drain tile. It drained a full month before his neighbors. Not only that, but it drained crystal clear water, whereas historically that would be a really dirty brownish gray water that would drain off of that tile. The neighbors, which finally started draining a month later, was as dirty as it could get. So we knew that a lot of his soil was washing away through his drain tile. So again, the anecdotes come. I mean, that's something that happens. Now, can you go to the bank and say, hey, my drain tile is draining clear water? Give me a bigger operator loan than I had last year? No, probably not. The bankers, they don't quite understand this yet. But
Maybe someday.
maybe someday,
With
but
these
we can,
environmental benefit discussions that are happening, that
as they should be.
yeah,
We are cleaning the system, and I think
right,
and I really realized this year, but I think at the very base of everything that we're doing, we're bioremediating. We're
right.
taking those chemicals and those legacy chemicals and those half-lives, we're shortening them in the soil so that our soil can hopefully return to a homeostasis and digest again. Because again, the soil is the gut. We need the
Back to the gut.
microbes in
Yeah.
there to help it digest.
Well, Zach, what a great take you have on it, and what wonderful experience you have to speak to this today. And really appreciate you joining us. I think that's a good note to end on.
All right.
Started with the gut, and we're going to end with the gut. And I hope everybody starts thinking about what they can do to keep their gut health and their soil healthy.
Yep.
And if someone wants to get a hold of you, your contact information.
Livingsoillab@gmail.com or www.livingsoil.net. And that essentially will take you right to that email or my phone or text. I field a lot of calls from people that are really new to this and they don't quite see the whole picture yet, but they're curious. And those are my favorite calls because I'm not here to give you a bunch of false hope, but I think that just about everybody that starts to wrap their head around all of this and then actually instigate and do these things starts to have more hope because we're growing the whole system, not just the crop anymore. We're growing the soil too. And I think it's time that we invest in our soils more than just a caffeine bang monster Red Bull energy drink to get that crop out of the ground so we can take it to the elevator. I think that we really can improve our future and we can extend our harvests as soon as we start to reinvest in our soils. And there's not a better reinvestment in my mind than microbiology.
Yeah, promoting life, isn't it?
Yep,
Promoting
exactly.
life rather than a death culture.
Yeah, exactly.
With that, Zach, thanks again. And of course, anybody can contact any of us at Elevate Ag as well, and we can connect you.
Thank you,
So
Jim.
thank you.